So KRAS is the most commonly mutated oncogene in human cancer. It's mutated in about 20% of all cancers. But frustratingly, this target has been incredibly difficult to drug directly. And while we're enthusiastic and optimistic about some of the ongoing efforts, like those you just heard about, to actually develop a drug that actually targets RAS, in the meantime, there's been considerable effort to look at alternative approaches focused on some of the downstream effectors of KRAS. And so this is probably a simplified version of what the KRAS signaling network looks like. But the problem with trying to do this is that KRAS turns on a very broad range of downstream pathways. Now, one pathway that has been focused on primarily is one of the most well-characterized pathways, the MAP kinase pathway, or the RAF MEK ERK pathway. One drug that's been explored most thoroughly in this setting are inhibitors of MEK. And there's reason for this. These are data produced by Cyril Bennis from the MGH cell line screen of about several hundred cell lines and about 100 targeted compounds. And actually, the most effective agent in this group of KRAS mutant cancers is actually MEK inhibitors. But still, when you look at what happens in vivo with a MEK inhibitor compared to vehicle control, sure, you get a significant reduction in growth, but as Neil alluded to before, you really get at best a cytostatic effect. And we really do not see true tumor regressions and cell death in any of the in vivo models treated with a MEK inhibitor alone. And this is important, and it really correlates, I think, with what we've seen with single-agent MEK activity in people, where we actually do see a very small single-digit response rate, but really a broad range of stable disease, albeit short-lived stable disease, but really overall a lack of efficacy as monotherapy. So an alternative hypothesis has been to at least build off this cytostatic effect by inhibiting MEK, and then trying to perhaps take out another downstream effector of RAS, or to at least find some synthetic lethal partner to go after. And as you can see, this is potentially a tall order. So to get at this, several years ago, we performed a shRNA mediated synthetic lethal screen, where we took a pooled library targeting kinases and other druggable genes and put them into KRAS mutant cancer cells. And then what we did was we split that population into three groups. We froze down an initial population to represent where we started. And then we treated cells with either vehicle or a MEK inhibitor, in this case with AZD6244 or selumetinib, for 7 days. And at the end, we then, using a sequencing-based approach, actually were able to measure the abundance of each of these sRNA constructs in each of these populations. And what we were looking for were not genes like this pink gene here that were universally toxic in the presence of vehicle or MEK. What we were really looking for were for things that specifically dropped out in the MEK inhibitor-treated population relative to the vehicle-treated population, like this blue gene here. We did this initially as a pilot study with 2 KRAS mutant cell lines, and what we focused on was the intersection of the top hits in each cell line. And this is what we found. And one thing that was at the top of the list but jumped out at us also for potential mechanism was BCL-2L1 or BCL-XL, which is a member of the anti-apoptotic family. So apoptosis in cancer cells is regulated by a balance of pro-apoptotic proteins like BIM, which was introduced earlier, and anti-apoptotic proteins such as BCL-XL that actually bind to and inhibit the action of these pro-apoptotic proteins. And when the balance here favors the anti-apoptotic protein, the cell survives. But when this balance is tipped in favor of the pro-apoptotic proteins, the cells undergo apoptosis. And so what we found here was somewhat interesting. So we heard about BIM earlier, and BIM is actually one of the key pro-apoptotic proteins regulated by the MAP kinase pathway. And it's been known for some time that if you actually treat cells with a MEK inhibitor, as in these last 2 lanes, you get a dramatic stabilization and rise in protein levels of BIM. And this is thought to be one of the key drivers of apoptosis with MAP kinase pathway inhibitors. But what was interesting is when we pulled down BIM, and actually looked at what was bound to BIM. We found that at the baseline level, you could detect some degree of BCL-XL binding to BIM. And when you gave a MEK inhibitor, even though you got a dramatic induction in protein levels, you saw that you pulled down a proportionally increased amount of BCL-XL, suggesting that most of this induced BIM was not actually free and perhaps was bound by BCL-XL and thus unable to drive apoptosis. However, if you treat it also with nigvidiclax, or formerly known as Abbott 263, which is actually an inhibitor of a protein-protein interaction and blocks binding of BCL-XL and BCL2 to proapoptotic BH3 proteins like BIM, you could actually ablate this binding interaction and now leaving this BIM free to hopefully promote cell death. And so our model is that at baseline, BIM levels are suppressed by the MAP kinase pathway, and any BIM that's present is actually bound and inhibited by BCL-XL for good measure. In the presence of a MEK inhibitor, you get this dramatic induction of BIM protein, but all of this BIM is actually bound and inhibited by BCL-XL. But now if you give a MEK inhibitor with an inhibitor of BCL-XL, you now free these increased levels of BIM to promote cell death. And that's what we observed in several in vivo models. These are 3 independent KRAS mutant colorectal xenografts. And what you see here is that in green, Abbott 263 or nigvidiclax alone has really minimal effect relative to vehicle. Again, in red, the MEK inhibitor really gives you primarily a cytostatic response. But now with the combination, we're actually seeing tumor regressions, as seen here on these waterfall plots, where each bar represents a tumor in a mouse. And this, at the time, was among some of the more impressive activity we've seen in KRAS mutant xenografts. But xenografts aren't entirely representative of human tumors. And so we turned to another model, that's a genetically engineered model with activation of an oncogenic KRAS in the lung in collaboration with Kwok Wong. And these tumors actually develop de novo. They have an intact tumor microenvironment, and they can be monitored by MRI. And so you can see that as these mice, given time after activation of this transgene, you see that the lungs of these mice are actually filled with tumor nodules. However, after I think this was just 1 week of treatment with the combination, you see a near complete regression of these tumor nodules. And actually, mice go from really having difficulty breathing to going back to kind of looking somewhat normal. And so we saw these very dramatic responses both in a model of KRAS alone as well as KRAS P53. And so based on these data, we're actually running a clinical trial right now funded through NCI-CTEP that's actually combining nigvidiclax with the MEK inhibitor trametinib. We're currently still getting toward the end of the dose escalation cohort, but after we reach our maximal tolerated and recommended phase 2 dose, we're actually going to open expansion cohorts in several tumor types where KRAS is frequent, pancreas, colon, lung, as well as a basket for kind of all other KRAS mutant cancers. And we've just amended this to now include NRAS mutant cancers as well. And so we've enrolled about 20 patients to this trial and are excited to get to our dose and see what happens. So given the success of that initial pilot screen with 2 cell lines and the potential promise of this combination therapy, we decided to expand our efforts. And we actually decided to do this screen again with now 6 KRAS mutant colorectal cancer cell lines and 6 KRAS mutant pancreatic cell lines. And we did this actually with a new library, which I didn't update on this slide, of about 11,000 shRNAs covering all kinases. The other thing that we did differently is instead of using selumetinib for a shorter period of time, we used a trametinib, which is a newer generation MEK inhibitor, at an exceedingly high dose. This is several times more than the maximal dose achievable in people. And the reason we did this is actually other groups, such as Neil Rosen's group, have found that when they've done similar screens, like our first version screen with selumetinib or an earlier generation MEK inhibitor, that actually many of the hits they get back are actually in the MAP kinase pathway. And one of the findings of a paper done by Neil and Pirulito, a postdoc in his lab, showed that actually newer generation MEK inhibitors actually could better suppress. So we decided to use trametinib, but also use it at an excessively high dose to really get only MAP kinase pathway independent hits. And so what happened? Well, it turned out 4 of our top 15 hits were actually still in the MAP kinase pathway. We had all three RAF proteins, or RAF genes, ARAF, BRAF, and CRAF, which is also called RAF1, as well as MEK1 itself. And so this was actually really puzzling because we intentionally designed this screen to avoid issues in the MAP kinase pathway. But this actually suggested that even with this kind of super physiologic dose of a new generation MEK inhibitor, we were actually still getting reactivation of the pathway. And that's actually what we saw. So I'll show you a little more data on this in a second. But this is essentially summarizes our findings, is that initially, out to 24 hours, you get very nice inhibition of phospho-ERK, one of the main downstream effectors of the MAP kinase pathway, and also RISC, which is a target that ERK phosphorylates. But after time, 2 days, 3 days, 4 days, you see that these levels gradually come back, and even more dramatically with an early generation MEK inhibitor, selumetinib. Now this is accompanied by a gradual activation in CRAF, as well as an intense increase in phosphorylation of MEK. So to try and figure out what's going on here, we actually looked to some work that we've been doing over the last several years, and I'll take a little trip back in time, about 5 years, some work we've done in BRAF mutant colorectal cancer, where we've seen a very similar phenomenon to this. Now, we're all familiar with the incredible efficacy of BRAF inhibitors in BRAF mutant melanoma, which is about 50% of melanomas, 60% or so response rates. But when colorectal cancer patients harboring the same BRAF V600E mutation were given one of the same BRAF inhibitors, vemurafenib, only a 5% response rate was observed. And so this really was a very striking difference in tumor type sensitivity to a compound in tumors that actually harbored the same mutation. Now, at the time this result came out, the prevailing hypothesis in the field was, well, colorectal cancers just are dependent on other pathways. They're just not MAP kinase pathway dependent. However, when we went to actually try and figure out what's going on here, we modeled what happens when you give a BRAF inhibitor to a BRAF mutant melanoma cells or BRAF mutant colorectal cancer cells. And what we found was quite interesting and suggested that perhaps the opposite was true. So in BRAF mutant melanomas, you can give a BRAF inhibitor and turn the pathway off and keep it off for an extended period of time. But when you give the same drug at the same concentration to BRAF mutant colorectal cancer cells, you inhibit it at a short time point, but the pathway gradually reactivates out to 48 hours. And this happens even if you replenish drug at every 24-hour intervals and even a few hours before lysis. So this suggests that actually perhaps another hypothesis is potentially also correct, or potentially correct, that we may actually just not be inhibiting the target pathway in these cancers, and that may be the issue. So through a lot of work that I won't summarize here, and this is very similar findings were produced by Neil and some colleagues at Memorial. What we found is that in the absence of drug, the primary mutant BRAF is the primary driver of MAP kinase signaling. When you give a BRAF inhibitor, you lose these ERK-dependent negative feedback signals, which are suppressing activation of the pathway through receptor tyrosine kinases in RAF. And what happens is you get a reactivation of the pathway through other RAF family members, most notably CRAF. So we tried to overcome this by combining a BRAF inhibitor with a MEK inhibitor to try and block this reactivation. And interestingly, even though this waterfall plot looks a little bit better, we have a 12% response rate versus a 5% response rate, we're still nowhere near where we need to be. And as I'll show you in a second, with biopsies taken right before treatment and at 15 days on treatment, what we found is we were inhibiting the pathway in everybody, but only by about 40 or 50%, and not anywhere near what we were inhibiting the pathway in melanoma with a BRAF inhibitor. So that led us to try a number of different strategies, targeting EGFR, which through some preclinical work we showed was responsible for this reactivation in some, but not all, BRAF mutant colorectal cancer models, and also combinations eventually of all three agents, anti-EGFR antibody, a BRAF inhibitor, and a MEK inhibitor. And to summarize a lot of clinical data, we just updated the final efficacy a few weeks ago at ESMO. And what we found now is a 32% overall response rate. Still short of what we see in melanoma with one drug, but at least now we're actually having some effect on this cancer. And really interesting is what we found is when we looked at these day 0 and day 15 biopsies, we found something very interesting that supported why these regimens may be more active. So in BRAF mutant melanomas with dabrafenib, a BRAF inhibitor alone, we get about 80 or 90% inhibition of MAP kinase signaling. With BRAF MEK, we inhibit the pathway in everyone, but again, only by about 40%. suggesting why the efficacy still wasn't where we needed it to be here. Interestingly, in BRAF EGFR, we inhibit the pathway in some people but not others, which is really consistent with the fact that some tumors, the pathway reactivation may actually not be driven by EGFR. Again, with with MEK and EGFR, we still are getting suboptimal inhibition. With the 3 drugs together, we're actually now getting about 60% inhibition, which again is frustratingly still short of where we are with melanoma, but at least getting into arena where we need to be. And this really suggests that the increased activity we've seen with these more complex combinations really is correlated with increased MAP kinase pathway suppression, again supporting the hypothesis that the MAP kinase pathway is a critical target in this disease. So frustrating though, even with 3 drugs, we're still not where we are in melanoma with 1 drug. And even more frustrating is despite the improvement in response rate, our median progression-free survival is only about 4 months. And so these patients are developing acquired resistance very rapidly. And when they do, what we found that almost everybody develops mechanisms within the MAP kinase pathway. We occasionally see MEK mutations, but we also see amplification of BRAF V600, KRAS amplification, KRAS mutation, and RAS mutation. This is very consistent with the data that Neil showed earlier. And so what we and we've also, seen RTK amplification. So what we see almost, virtually almost every time is that these mechanisms lead to MAP kinase pathway reactivation. But I think what's very interesting about this is that almost, despite the fact that we're using a high-level combination with a MEK inhibitor, almost all of these resistance mechanisms are actually upstream of MEK, with 1 exception. And that's very unusual because in most systems, you expect to see resistance occurring at the level of the target or downstream of the target. And so this suggests some very interesting biology in this pathway. And this actually goes back to one of the first papers I published as a postdoc, where we treated BRAF mutant colorectal cancer cells with a MEK inhibitor until they began to grow and were thoroughly resistant to the drug. And what we found was amplification of BRAF V600E, something we've now have found quite commonly in patients. Interestingly, again, what we found was that with a MEK inhibitor now, instead of having very potent suppression of MAP kinase pathway, we had about a 1 to 2 log shift in the IC50 of this drug. And what was actually striking about the pharmacology here was that if you looked at the IC50 of the MEK inhibitor in parental cells in yellow versus resistant cells in red, you have this nice large shift. However, if you actually co-treat these cells with a subtherapeutic dose of a RAF inhibitor, just enough to get these elevated levels of phospho-MEK down to what they are at baseline, you can actually now reduce the potency of this drug back to normal levels. And actually, this is actually the biochemical data from the abundance of phosphoric. You actually can shift the potency to inhibit phosphoric back to normal. And so what this suggests is that there's a very interesting pharmacology here where if you have excessive upstream activity, you can actually almost override the ability of the MEK inhibitor to inhibit. And the model for this is that at baseline in a parental cell, you have a pharmacodynamic relationship where about a 50% inhibition in MEK leads to about a 50% inhibition in ERK. However, in these resistant cells, what happens here is your levels of phosphorylated MEK are increased about 6 or 7-fold. And what ends up happening here is when you compare the relationship, you have about 3 times what you need for maximal activation of ERK. And so if you then treat with a RAF inhibitor to slowly bring down levels of phospho-MEK, you find that the IC50 is the same. But if you look at the absolute levels, to achieve this 50% point, you need to actually inhibit the overall levels of activated MEK by about 90%, and that takes about an extra log of drug. And what happens now is you get this shift in the relationship where you actually have to inhibit MEK to you have to actually inhibit MEK considerably more to get to about 90% to get a 50% reduction in ERK. So this suggests that what's going on here are these resistance mutations are essentially increasing the signaling flux through MEKs, hyperactivating to the point where the MEK inhibitor can no longer keep the pathway blocked. And I think this is really unusual because when we think about drugs, if they have a name like a MEK inhibitor, we think it's a solid block at that level. And I think that from what Neil showed, and I think from some of the data here, there's a very dynamic signaling pathway, and there are very complex feedback loops, and we can't just assume that we turn the pathway off completely. What's interesting, though, is that in almost all of these models, in fact, pretty much all of these models, we found that the ability to shut down the MAP kinase pathway, and in most cases to restore the majority of the sensitivity could actually be overcome by ERK inhibitors, just stepping one step downstream and avoiding this unusual pharmacodynamic relationship where you have MEK hyperactivation. So we decided to try this in the KRAS model and see if the same principle applied. And indeed, when we now treat with an ERK inhibitor, this is a Vertex tool compound, what we see is actually an induction of phospho-ERK, which is what you see with some ERK inhibitors. But if you look at kind of what's a universal target, an ERK target, phospho-RISC, you see that rather than getting this feedback over 96 hours, you're able to maintain and suppress levels of MAP kinase pathway activity. And this is despite still having the same feedback of CRAF activity induction and induction of phosphorylation of MEK. This is not just unique to the cell line. In our entire panel of 12 cell lines, we looked at the ability of selumetinib and trametinib to keep the pathway suppressed over 96 hours. And we found that compared to the 24-hour time point, there is a statistically significant increase in the levels of risk phosphorylation by 96 hours. But that's not the case with an ERK inhibitor. So we wanted to try and really model what's going on here and see if this hypothesis that we had developed in studying BRAF mutant colorectal cancer actually applies here. And so what we did was an interesting experiment where we actually pre-treated cells with trametinib out to that 4-day time point where we typically stop our westerns. Then we washed the drug off and actually added back a concentration curve, which we then kept on for about 2 hours. And we did this to pre-treated cells as well as to cells with no pretreatment. And the idea here was to let all of the changes that occur during this adaptive feedback and then assay what the effect of that is on the potency of this drug to inhibit the pathway. And what we found is about a log shift in the potency of trametinib and about a 1 to 2 log shift in the potency of selumetinib to inhibit phospho-RISC after a 4-day pretreatment with trametinib. And as you can see, the baseline MEK levels, phospho-MEK levels, are massively induced at this time point compared to the non-pretreated cells. Again, we tried another way of modeling this by actually just trying to increase the flux through the pathway by really just expressing high levels of mutant KRAS. And again, what we saw here is just under a 10-fold shift in the potency of selumetinib and trametinib in this KRAS model, but no shift in the ability of the ERK inhibitor to turn this pathway off. We think that certainly this I think supports that increased pathway activation is at least one of the mechanisms that allows MAP kinase pathway reactivation in the presence of a MEK inhibitor. But there are also some other potential mechanisms that we're exploring right now, some of which may involve some of the downstream negative regulators that for which ERK regulates their transcription. Those can actually be used as a readout for MAP kinase pathway activity. And again, when we look at transcript levels, we see the same effect where for selumetinib and trametinib in red and blue, we see this gradual reactivation of the pathway over 96 hours, but we see maintained suppression of transcript levels with the ERK inhibitor. Importantly, this ability to produce more sustained inhibition of the pathway actually seems to translate into better efficacy. These are long-term in vitro assays where cells are cultured with drug and media replenished every 3 days. And what you can see is certainly compared to selumetinib, but also trametinib in all of these models, you get a clear and statistically significant a stronger inhibition of cell viability than with the MEK inhibitors. And in some cases, we see really primarily a cytostatic effect, some not quite as strong. But in some cases, we actually see some reduction in cell number overall, suggesting that perhaps even with really strong and optimal MAP kinase pathway activation, we may actually lead to cell death in some models. And this might be consistent with a rare patient or two who does have a response to this drug. Importantly, MEK inhibitors have been involved in almost every combination trial we've looked at for KRAS mutant cancers. And we see that replacing the MEK inhibitor with an ERK inhibitor gives perhaps more optimal suppression of cell number with palbociclib, a CDK4/6 inhibitor that's actively being investigated in combination with MEK inhibitors in KRAS mutant cancers, as well as navitoclax, which is the BCL-XL inhibitor that's part of our trial. So when we take a step back and look at why have we had such little success with our downstream targeting efforts in KRAS mutant cancers. When you actually do a search for RAS and MEK inhibitor, you see that there's about 31 studies that come up. And there have been a tremendous number of trials that have combined a MEK inhibitor with a PI3 kinase inhibitor, with a CDK4/6 inhibitor, with a BCL-XL inhibitor. And time after time again, so far, the data has been disappointing from these trials. And so is it possible that one of the reasons that these have failed is that we're actually not producing sustained inhibition of this pathway like we think we are, similar to the way that we weren't inhibiting the pathway with a BRAF inhibitor in BRAF mutant colorectal cancer? And so if we actually should be, before we start combining with other agents, should we actually concentrate on a strategy that actually more optimally inhibits the backbone pathway that we're targeting? And again, some evidence that this is true. These are actually some mice treated for either 3 or 4 weeks with a MEK inhibitor-based combination. And you see that while we have pathway suppression at these early points, by a few weeks later, you have, again, reactivation of this pathway pretty consistently in the remaining tumor cells. And so while we haven't really yet shown clinical evidence of this in people, this is something that I think we really need to investigate. And we do have built in some on-treatment biopsies in our trial that we're hoping to evaluate once we get up to full doses to see really what degree of pathway suppression we're getting. And so I'll quite early stop there. So I think that despite our, the somewhat, setbacks and disappointments in the early clinical efforts, I do think that combination therapies for KRAS mutant cancers really do remain a promising approach. But I think that that these data suggest that the primary backbone we've used for these trials, or MEK inhibitors, really may not produce sustained MAP kinase pathway suppression. And that is one, at least, potential explanation for why these trials may have failed. So I think that before we try additional MAP kinase inhibitor-based trials in KRAS mutant cancers, I think we really need to focus on coming up with a strategy that we're confident produces more optimal pathway inhibition in these patients. And while there are several potential approaches to this, pan-RAF inhibitors, ERK inhibitors, or other, I think, or perhaps newer, even next-generation MEK inhibitors, I think that our data at least suggests that ERK inhibitors, which are currently in clinic right now, may offer some advantages over MEK inhibitors by producing more sustained MAP kinase inhibition and actually being less susceptible to this upstream reactivation of the MAP kinase pathway. And I think that these data warrant that ERK inhibitors should be explored as potential kinase backbones to inhibit the MAP kinase pathway in KRAS mutant cancers. And so I'll stop there and take any questions. A lot of people to acknowledge for this work. Leanne Aronian is the postdoc in my lab who produced most of the recent data with ERK inhibitors. A lot of the folks here, Jeff Engelman, some of this work was actually done back in the day when I was a postdoc in his lab. And a tremendous number of collaborators in pathology and in the clinic. Some collaborators at the Broad who've done some work in the resistance, the BRAF colorectal resistance data, and my funding sources. So I'll take any questions.
The rapid recovery suggests some kind of an adaptive mechanism of resistance.
Right.
Are you looking into that now?
Yeah, so certainly I think that's what we're seeing here. I think that interestingly, there's been, Neil and Scott Lowe's group just published a paper showing that in KRAS mutant lung cancer, at least, there's a primary RTK that drives it, much kind of similar to the EGFR model in BRAF mutant colorectal cancer. Some other papers have shown some similar findings. A paper from a few years ago actually implicated some of the HER family proteins. But interesting, when we've actually looked at this to try and see if we can hone in on a single RTK, at least in this panel of pancreatic and colorectal cancer cells, we find that it's quite variable. When we look at RTK arrays, we see some mild induction of some, but not others. When we look at actually test functionally with different inhibitors of RTK pathways, we sometimes see one that has a little effect in one cell line, but we don't see a universal model here. And while that may be the case, or perhaps more universal in something like lung cancer, at least in these models, we aren't finding a consistent an RTK to go after. So what we do think is that the loss of these ERK-dependent transcripts, such as Sprouties and DUSPs, lead to this kind of reactivation, perhaps promiscuously by RTKs, but we do think it occurs at the level of RAS. And I think the kind of the ambiguity of what the exact mechanism is, or the lack of a clean offender, might make it hard to try and target that in addition to a downstream point. And so we've actually tried to just look at if there's a way to cut the pathway off kind of regardless of what's going on up top.
Couldn't agree more. Let me make an extra comment on that and then ask you my question.
Sure.
I was shocked. Now I'm going to say the data is wrong so I can say it's Scott's work, not my work. I was shocked that he got so much FGFR because I think it's going to be, we did work, you probably know, with Surat Chandalapati in breast and other tumors. It's a lot of kinases. And I was surprised to see the FGFR. An interesting thing from that paper is he found FGFR in pancreas and lung, but not colon. So there's certainly going to be lineage. But I will bet, and you will join me, I'm sure, that when you give FGFR plus MEK-ERK, you'll get another kinase when you're inhibiting the
I think
FGFR.
there's no doubt about that.
And which gets to my real question. First of all, I agree with this completely, and I think it's fantastic work, the best work in the field. Of course, if I disagreed with it, it would ruin my career. But I think you left out the elephant in the room, which I often leave out. We give an inadequate amount of ERK inhibition, and we have dose-limiting toxicity due to ERK inhibition. So we have to grapple with that question. My only answer, as you know, is intermittent therapy. Not that I know that it works, but it's going to have to. Current thinking, I would say, I have no other answer to that. In the RAS tumors, they missed the opportunity. In the RAF tumors, you can give a RAF inhibitor, and that, as you well know, prevents the toxicity. And you can probably go up even higher with MEK. But with RAS, I don't have any other solution to hoping it will work in pulse doses. Do you have any other?
I think you're absolutely right. In fact, we've actually moved to pulse dosing, Q2-week dosing in the CTEP trial that I showed. I think one of the reasons we've had, well, one of several reasons there's been so much success, I think, has really been kind of, something that you've showed in BRAF mutant cancers is that you can crank up the dose of the BRAF inhibitor. You inhibit the pathway in cells that have BRAF V600, but you don't have as much of an effect in cells that have wild-type RAS. And in some cells with a degree of RAS activity, you might actually get induction of the pathway. So that was one of the, I think, the really interesting things about the BRAF MEK EGFR trial in BRAF colon is that we gave the BRAF EGFR, it was fine. We gave the 3 drugs together at full dose, fine. We took the BRAF inhibitor out and tried to give EGFR MEK, and people's skin just melted off. And we had to dose reduce. It was still not tolerable. And it's just amazing that this incredible biology of this pathway where the paradoxical activation by the BRAF inhibitor really is able to kind of suppress the otherwise dermatologic toxicity that you see with MEK. But in RAS, we lose that therapeutic window there. And I think one interesting thing we might try and look at is, I don't know if these adaptive feedback mechanisms are stronger in the tumor or stronger in your normal cells like skin. And it'd be interesting to see if we actually might be getting greater inhibition in the skin than we are, or in other normal tissues than we are in the tumor. It's certainly, I think, likely that that's the case. But yeah, the therapeutic window is an issue. It's a huge issue. And I think the intermittent idea is you hit them hard, kill them, and then don't have to, at least don't have to worry about that subset that you killed anymore. And you'll have to deal just with what's left.